They're multiple choice in format. If you come across one of those that you don't know how to answer, take the opportunity to get that. Your homework is due by the end of today. Usually by Wednesday, like 10 or 15 people in the class have already completed it. I think 3 people have done it so far. So it's like, you know, you're kind of getting in there and getting comfortable and getting on cruise control. I get it. But just remember that the initial post is due by the end of today if you want to get full credit for that. It's a little bit more descriptive in terms of what it wants you to do. It asks for 3 arguments for and 3 arguments for against. And so it's a little bit clearer exactly how much detail you ought to include in that. I think that's it as far as structural issues, unless there are any questions. Yeah, one of you sent me an email saying that you weren't getting the emails that I sent out. So I'll work on that. I don't know what the issue is there because I just tell it to email everybody enrolled in the course. So we'll figure that out. I put the last 2 lectures on YouTube. I think I'm going to continue to do that. I don't know if it's helpful for you or not. But it really requires very little effort on my part, so I'm going to do it regardless of whether or not it's helpful for you. Like, I forgot to have it triggered, and it took me, 10 seconds just right now to get it fired up. So, yeah.
I clicked on the link, and it didn't work. It just brought me to the page it said before.
Interesting. Anybody else have trouble clicking on the link? Okay. I'll get the link in addition to in an email. I'll post it on the Canvas page, and that might help with that. And then you could also, if you just worst comes to worst, just copy it and paste it in the and that should help. All right, cool. Let me pray. Father, I thank you for how our weeks are structured. I know it's just seems like such a small thing to thank you for, but Father, I know that I need it. Father, I need the typical routine and the typical structure of our week because, Father, I'm so weak, and I know that's true for a lot of us. And I thank you for the opportunities you've designed for us to take rest. And I know you designed it that way because your word teaches us that. Father, help us to do that well and be responsible and be good stewards of our structure and our week, that we might have the energy and the focus and the diligence that we need to work hard for you when it's time for that. And Father, as we sit here this morning and into this afternoon, Father, I pray that you would help us to think clearly and think well. And as we spend some time discussing, Father, I pray that you would reveal to us what you would have us to know, and that you would reveal to us what we can know about you from studying your creation. We ask this in the name of Jesus. Amen. So we left off here last time, right? On Monday, showing you some of the taxonomy. And basically to really quickly review the setup for this, this was an answer to the question, do eukaryotes evidence universal common descent well? And the basic answer to that is no, and there are a number of issues. One being this polytomy, which whenever you see a polytomy, which happens a lot when you try to take this many organisms and put them into what you would call a phylogeny, representing the evolutionary history. Because you're just looking at way too many organisms, and I would argue you're looking at organisms that don't actually share ancestry, and that's why it's so messy. Okay, but we've got this polytomy where one branch splits into more than 2. Certainly not the way we see speciation work. And then on top of that, we have a number of issues in terms of where there's just a great deal of debate in terms of whether we should group these organisms together at all. However, there is still some value in having these groups and in grouping this way, because as you classify and organize living beings, you realize that there are certain themes and certain structures that work well. And then you start to pick up on why they work well, and you start to understand the importance of why organisms are structured the way that they are. So we'll save our discussion of excavata for a little bit because we're going to focus in on a parasite from this group. Chrome aviolata, this is just kind of a conglomerate group, and you'll see there's a lot of disagreement about the relationship, whether these should all be grouped together or not, but there are some shared features. Great deal of diversity in here. The overwhelming majority of photosynthetic organisms on Earth are in this group. Dinoflagellates and diatoms make up the overwhelming majority of all photosynthetic organisms on Earth, which, without photosynthesis, you don't maintain oxygen levels at the right rate in the atmosphere for human life to work well. So it's kind of important. Rhizaria, again, we're not going to talk a great deal about this. There are some really interesting marine forms that contribute a great deal to the sediment, and you can find some really neat fossil remains of them, but it's a bit beyond what we have time to discuss here. Yeah?
It's kind of a random question, but how would an evolutionist argue how plants who depend on carbon dioxide and animals who depend on oxygen evolved from a common ancestor? Because they do have a
Right, but you'd have to go back a long way. So that actually brings up a good point. So if you see land plants are here in Archaeplastida, and then animals are in here in Pisthocanta. So to go back to the original ancestor of plants and animals, you have to go all the way back to the original eukaryotic organism, which is understood to have existed about 2 billion years ago. And in 2 billion years, you can generate a great deal of changes. But yeah, I mean, it's certainly not a recent ancestor between them. There are a ton of differences and issues you have to work out to relate plants and animals, other than just, what's going on biochemically. What do they use? How does respiration work? Yeah. But so Archaeplastida, we'll talk about these, and we're going to focus in on a couple of groups in here. Some of these we won't deal with a lot, actually, until we get to plants, even though they're protist. Some of our charophytes, chlorophytes more generally, are green algae, because this is the group that land plants are supposed to have come from. So traditionally, all of the algal groups were grouped together: red algae, green algae, brown algae. But some things have changed to that. Brown algae is now in Chrome Aviolata. We'll talk a little bit about Amoebazoa, just about their forms. They're really, really weird and really strange organisms. And there are some of them that look a lot like fungi and used to be classified as fungi and are no longer. And so it's a good reminder of just this idea of convergence. I showed you last time, right? Convergence versus homology. Did I show you that? The difference? We'll talk about that some more. And you'll love it. I'll love it. And then finally, we'll talk about Pisthocanta, but really not until we get into fungi and animals. We're not going to talk about it a great deal this week. Yeah.
So looks like there's another polytomy in that branch for Chrome.
Right here. Oh, Chrome Aviolata. And it's right here. Yeah, you've certainly got a polytomy here.
And that's just as problematic as the original.
Oh, sure. Yeah. Anytime you see a polytomy, you've got a problem there. Something is unresolved. And if your relationships are unresolved, it's an indication that even with the amount of data we have on them, we can't root them all back to a single ancestor. Yeah. Okay. So here's some examples of protists in these particular groups. Dinoflagellates, Chrome Aviolata, really complex organisms, oftentimes having this really complex armor, usually made out of cellulose, which is the same thing plant cells use to make their cell walls, and usually having a flagellum coming out, so they're fairly mobile. Some of the most abundant photosynthetic organisms on Earth, in enormous variety in shape and form. I mean, you could look at diatoms for the rest of your life, and you would never run out of different varieties and shapes and colors even. Just incredible diversity. Here is a wave right here in this image, and there's the bioluminescence from these dinoflagellates. And it's incredible. And then there's an interesting question here of what on earth are they using this bioluminescence for? Because a lot of organisms that do this, that flash, that actually create light, use it for communication. And yet we've got single-celled protists that, I mean, what on earth would they say to each other, even if they were talking? And yet they have this ability to generate light. And it's really fascinating. And then other organisms can eat them, and do, because they are some of the most abundant photosynthetic organisms on Earth, which basically means they're making the original biomolecules. So almost everything eats them and can actually incorporate that bioluminescence into their tissues, maintain some of that. Really cool. And then we could also take the genes that are associated with that and put it into the skin of mice and get mice to create that bioluminescence. I mean, I don't do that, but other people do, and it's pretty cool. I don't know the value of it, but it is. I mean, it's pretty neat. Yeah, we have these. Like safety knobs that we put on our doorknobs, so like our 2-year-old can't get to places where she shouldn't, and they glow in the dark. And I'm like, what's the value of that? I mean, when you're walking, I mean, your house has to be really, really dark for you not to be able to see a doorknob as you're walking around at night. And do you really want a kid that can't open this to be able to find it more easy? I just don't understand. But it does glow in the dark, which is exciting, I guess. Paramecium, we talked about this in context of the example of sexual reproduction that we saw, which again, as a reminder, a number of protists are capable of reproducing sexually and often do in accordance with when environmental conditions are going to get harsh. It's a way of kind of scrambling the deck, mixing your deck with somebody else's to maybe get some novel, unique combinations that might be more equipped to handle the environment than you are. Plus, it tends to also go along with forming cysts and riding out really harsh conditions. Straminopiles, again, another chrome alveolate, just demonstrating some of the incredible diversity within this group, which, with the amount of diversity in here, probably gives you some idea of why these relationships are so unresolved. Because you're probably looking at just a number of different unique creations that you're trying to root all of them back to an ancestor that never existed. Diatoms, along with dinoflagellates, again, these are the most abundant, common photosynthetic organisms on Earth. Have you ever heard of diatomaceous earth? Even if you've never heard of it, you've used it. It's in almost everything, lotions, toothpastes, makeups. It's just everywhere. And it's a really, really fine, sandy sediment. I mean, just incredibly fine. It's like the best sand you've ever held. You know, if you like to build sand castles, or you like to take sand and just spread it in people's hair because you know how difficult it is to clean that out, right? I mean, it is just incredible, and it's made from the remains of diatoms. And so diatoms will die, whatever's left over from their test, their shell will settle down, and then as it breaks down, it just leaves you this really fine, nice sediment. Okay, so here's the life cycle of brown algae, and the reason why this is up there, why it's in the text, and why I maintain it is because it's very interesting. This life cycle of brown algae has two different parts to it. It has a part where the cell, the major organism type, is what you call haploid, meaning it has a single copy of the genome, and then a portion of the life cycle that is diploid, having two copies of the life cycle. And you call this alternation of generations, where one generation is diploid and one generation is haploid. And what's interesting there, and so the gametophyte is haploid, the sporophyte is diploid. This is the way plant life cycles are functioned. And so what's interesting is the idea is that plants come from green algae, but the algae with the most plant-like life cycles are actually brown algae. And so this would be another example of convergence, where you've got this really unique system in place. You know, you can have convergence with structures as well, but it doesn't come from common ancestry. So brown algae and plants don't have this kind of a life cycle because they share a common ancestor that had that life cycle. It's just something that they have. So then it begs the question, well, what forces would cause that to develop in the first place? Or is it a remnant of the original design? And I think a lot of these examples of convergence are better explained, if your view of origins will allow it, are better explained by a designer. that actually set this in motion, because it's hard enough to think about, well, what would have originated this in the first place? What's the value? What's the driving force behind that? And then to have it appear multiple times through convergence. So a lot of these examples of convergence are really wonderful examples of a designer, of having certain things that were set in place. All right? We'll talk a little bit more about this when we get into plants. We'll leave it at this, just say brown algae. This is another chrome alveolate with a really complex life cycle, a plant-like life cycle. So here are a couple of organisms from Rhizaria. And Rhizaria, these tend to have really, really well-built, really complex shells. And so they tend to preserve really well. You find a ton of them in the fossil record because they have really hard, really rigid, really ornate shells. So when you find them in a rock deposit, it's very obvious what you're looking at, even to somebody who's not trained to find particular organisms. And the really hard shell tends to survive well through that process of fossilization. Here's another one. These are foraminiferans. Forams for short. Again, really nice, well-built, ornate, hard shells. And so here is fossilized remains of a radiolarian, a member of Rhizaria. And again, all of the members there make these really complex, ornate, hard shells. And this was actually described and imaged using electron microscopy. Really small structure, but incredible detail. Yeah.
Do we see a lot of change between the fossilized remains of these organisms versus what's present today?
Not really. Yeah, not really. I mean, you can find species of forams and radiolarians that have, what are supposed to be hundreds of millions of year old fossils that, I mean, they're virtually unchanged. Yeah. We call these organisms living fossils, where you have something that basically appears structurally identical to the way it did, when it first appeared in the fossil record, which is always interesting because the Earth has changed a great deal through that time. Regardless of your view of origins, the Earth has changed a great deal. And then so there's the question of why does that form stick around, right? What is it about that form? Is it really well developed? Or is it just something that they're biomechanically incapable of developing something else?
That's interesting because I would assume that these organisms would change a lot.
Well, I mean, they're really small, and I mean, they can move into a lot of different environments, so there's a lot of potential for new environments, and new environments tend to contribute well to novel shapes and changes. Yeah. Okay, any other questions? So that's it for Rhizaria. I told you we're not going to talk a great deal about them, and then we'll get into Archaeplastida. So Archaeplastida, again, this is the group that land plants are in. And with these 6 supergroups of eukaryotes, you've basically abandoned this system of grouping protist eukaryotes that aren't animals, fungus, or plants separately from everything else and in their own category. The supergroup completely abandoned that. And it takes all eukaryotes and puts them all into 6 supergroups. And so now there are some of those protists that are more like plants than other protists are, right? And there are some of those protists that are more like animals than other protists are. And so this supergroup Archaeplastida includes plants. And so you would imagine that the organisms in here are going to be more plant-like than some of your other protists are. All right? And so a good example of this is Volvox. Volvox Aureus is the scientific name. This is a really interesting eukaryote in that it exists as single-celled organisms and yet will develop really complex colonies. Well, there'll be some amount of division of labor, which is basically one of the definitions of multicellularity, where you have multiple individuals present and there's some division of labor. And so it's actually really interesting, and they'll make these complex colonies, and those colonies will start to reproduce. It's like when the colony gets too big, some individuals will actually leave that are reproductive individuals that will go and start a new colony. It's really interesting, and you find this a lot in a lot of protist groups. And the idea here is that this should give us some clue as to how multicellularity started in the first place. Because if your view of origins says that all of life is going to root back to a single eukaryotic ancestor, that is single-celled, you have to come up with some explanation of where multicellular forms came from. Some drive for why that would happen and some way of how it could have happened. Yeah.
Are they all not able to reproduce? So in some way,
Well, they don't all reproduce. When they actually get in that colony, some of the individuals will reproduce and form new colonies, and others do not, because there's some division of labor there. Some individuals are protecting the colony. Some individuals are helping to keep everyone together, and some individuals then reproduce, become reproductive individuals. Actually, is a really interesting. It'd be very similar to a biofilm in bacteria. Very similar functionally to what's going on there, although it's not anchored to something, it's free floating. But a lot of similarities to a biofilm. All right, we got a couple more slides before we have our lecture break, so push hard. I'll push hard, you push hard, and we'll get there, okay? And then we'll have a lecture break. All right, so here is a chlorophyte, which you could probably imagine what type of algae this is. What's your guess? Wonderful guess. This is green algae, right? You can tell because of the way it is, right? Green algae. And so Caulerpa toxifolia. And so the interesting thing here is you have a very plant-like form. And so you look at it, and it looks very plant-like. It's got blades, which are equivalent to plant leaves. It's got a rachis, a shaft, which is equivalent to a stem or that shaft of the leaf, and a very plant-like structure, and yet it's a single cell. It's a single cell that has all of this complex morphology. When you go into it, there's no divisions. It's just all one big cell with a whole bunch of nuclei. And so you have this really complex form. And there are a few reasons for thinking why this would be the type of algae that land plants came from, if in fact land plants did come from algae. And a lot of it has to do with the pigments. What pigments do you find in? They're all photosynthetic. Even plants that eat other objects, like the Venus flytrap and the corpse flower, even plants that eat other objects are still photosynthetic. Right? And so they need certain things that are going to absorb that sunlight. And what those things are pigments. And the pigments of land plants are most similar to the pigments of green algae, which is why land plants tend to be green. Green algae tend to be green. Okay? Because they have those same pigments that absorb basically the same wavelengths of light and reflect the same wavelengths of light. And that's one of the major reasons why this is the group of algae that land plants are believed to have come from. There's also some more genetic similarity. But there are some interesting things. First of all, green algae do not have the alternation of generations like land plants do. Brown algae do, but green algae do not. And there's some interesting issues, which we'll talk about that when we get to plants. Keep pushing. We're almost there. All right. And then you have amoebozoa. So amoebozoa, just a group of organisms where individual cells tend to move using pseudopodia, right? We talked about that the three modes of locomotion: cilia, pseudopodia, flagella. Okay, and so we've got these group of organisms that tend to move using pseudopodia, or what you would call cytoplasmic streaming, basically just rolling the whole gel of the cell in a particular direction, and the whole thing will move in that direction. And so a lot of these exist as single-celled organisms, don't ever form complex colonies. You could just find them under a slide. Some of these are massive. You'll probably see one in lab that a living amoeba will take up the entire field on medium power. Just a really big organism, almost big enough to see with the naked eye. If you've got really good visual acuity, you probably could. And then you have a number of forms in amoebozoa that are colonial. And so you've got two different types of slime molds. And this is our group that I mentioned used to be grouped with fungi, but now have been separated because of some genetic relatedness. And so now the idea is that this kind of an existence where single cells can accumulate, forming these large masses and then forming fruiting bodies where some individuals can actually leave as spores, that is another example of convergence. Okay, that this complex pattern of going from free living, single celled, to forming really complex colonies growing on some material that they're eating, to then forming a fruiting body where some reproductive individuals can leave as spores, which you find in almost all of your fungi, is now, again, another example of convergence, which you should think of as just another example or another opportunity to ask, where did that come from in the first place? What's the drive to get this to originate, or is this a remnant of the original design? And is this just a design that's really works well? And it really does. And then we actually use some of these organisms, slime molds. So this is a plasmodial slime mold, a little bit different in structure in the original form from this. Cellular slime molds. Cellular slime molds tend to spend a lot more time as single cells, kind of just going through a section of asexual reproduction, just going and generating more and more and more single cells, and then something triggers a more complex formation. They'll form these colonies and again get a fruiting body. Very similar to what you find in fungi, but they're not believed to be closely related. To get to the relative, the most recent common ancestor of slime molds and fungi, you'd have to go all the way back to your original eukaryotic organism, believed to have existed about 2 billion years ago. So they are just as closely related to fungi as they are to us, is essentially what you're saying, even though there's an enormous amount of similarity in their structures. So how would you explain that? What's that term? When you get two similar structures, but it's not from common ancestry. Convergence. I love it. Okay, let's have ourselves a lecture break. Okay? You need it. And so I want you to think about this, and I want you to spend a little bit of time just kind of thinking about it on your own, and then I'll open it up for you to talk with those around you. But I want you to think, okay, so we've got all of this diversity. We've basically given you a survey of the diversity of protists, right? And we've talked about that they're no longer believed to be a kingdom on their own, right? That they're just, kind of spread around among all the rest of the eukaryotes. So I want you to think about this. If you had to come up with 5 characteristics of a protist, what would those characteristics be, right? Keeping in mind that, yes, I know we can't group them apart from the rest of the eukaryotes, but if you were to say, if somebody were to ask you, what is a protist, how would you answer that question? By giving them 5 characteristics and say, this is what is a protist, okay? I want you to spend a little bit of time thinking about that. I'll open it up to those that are around you, and then we'll spend some time together. So take about a minute, starting now. Just think about it. All right, go ahead and take some time, talk about it with those around you. Come up with a list of 5 characteristics that protists have that make them a protist.
I got they can function either alone or multiple.
Okay.
Some other eukaryotes. Any other.
I got 4. They're eukaryotic. They're single celled. And high diversity of organisms, which I don't know if that's a good description. How would it?
Yeah, because there's high diversity of other things too, like you had bacteria, say, I don't know, there's multiple species of.
Well, high diversity, when you think what it means is that very a plant is
Yeah.
and animals
Yeah.
how do you word that properly? Can reproduce sexually or asexually? That defines although many other plants.
Yeah. Number 5.
The way they get energy, they were talking about both.
Yeah. Or they can be. So, I mean, if you think about it too, like, not.
All right. What do we got? And by the way, it's really nice when your list of features separates protists off on their own, right? So it'd be nice if your 5 features are unique to protists, right? And so I heard some discussion of, well, let's call them eukaryotic, right? Well, that's good. That is something that all protists are, but it certainly is not unique to protists. Yeah.
Okay, my brain's just like getting lost in the taxonomy. So we have archaea and bacteria
Yes.
and
Eukarya.
Okay.
Yep.
Are protists underneath eukarya?
Yes. Yep.
Okay.
And so let me do this for you.
I'm losing.
Okay. And so this is what we're dealing with. So we've got this this phylogeny representing the evolutionary history of life on Earth, right? And so the idea here is that bacteria are the most primitive of living organisms. That they are the most like this original ancestor that then gave rise to the rest of them. And the idea there is that they're fairly similar to some of your alpha proteobacteria, which happen to be the most primitive of our bacterial forms. A lot of those forms are anaerobic, meaning they like to, work in the absence of oxygen, which is the idea of what the original Earth would have been like. And then from here, you get one lineage of this group that then splits into archaeans, many of which are extremophiles, but not all of them, and then also splits into your original eukaryote. And then this original eukaryote, through some really huge mess that we haven't resolved yet, then gives rise to all of our eukaryotic forms, including protists.
Okay. And then prokaryotes are bacteria and archaea?
Yes. Yeah, and this whole thing... Okay. So this whole grouping, if you were being a good, what you would call a cladist, right? You're being a good cladist, which means that you only name things in such a way that represents their evolutionary history. You would never ever use that term prokaryote. Because you only use terms where all of the descendants and the ancestor are all in that group, right? And so here's the deal with prokaryotes. Is this ancestor here? You would want to classify as a prokaryote, but then some of its descendants are in a completely different group altogether.
Oh, okay.
So you would not want to use that.
Okay.
But it's such a valuable classification, it's such a valuable taxon that you can't help but to use it. But you don't want to, and so you're torn, right? You wake up in the morning, you don't know what you're going to do. You don't know if that day you're actually going to be okay using the term prokaryote, or if you're not, because it doesn't actually represent evolutionary history, but it's so valuable, right?
Okay.
And living life is really complicated. Actually, I don't think you'd worry all that much about it, but it is a real issue. And so we've got a big mess. So let's deal with it. What is a protist, right? What do protists have that other eukaryotes do not? Give me 5 of them. Only 5. Right? I'm not asking for 100. Yeah.
You said the majority of them are colonial, or at least they like to be around groups.
I don't know if the majority, but many of them are. Yeah, many of them will associate with other individuals. Although, basically, all of your plant, not all of your fungi, but all of your plants, all of your animals, they are like colonial and the next step, right? They are multicellular. So it's a good feature, but it's not unique to protists. Yeah.
Cyst formation?
Cyst formation. Okay, and a lot of them do, and a lot of that do that along with sexual reproduction, forming cysts. But that's not unique. A number of plants do that, and even or not a lot of plants. A lot of fungal species do that, and many animals do that as well to ride out really harsh conditions. Yeah. Non-vascular. That is true, but you've got a ton of plants that are that way as well, and basically all of your fungal and even some animals as well.
So like single-celled organisms?
You have that would get you close. That to say that at least a portion of their life cycle is lived as a single cell, but we've got a lot of fungal species that do that as well. But that would get you close. Man, that one's really close. I will tell you, this is an impossible task. You cannot do it. You cannot come up with 5 characteristics of what is a protist, because it is a horrible way of classifying organisms. So there's value to prokaryotes, even though if you're a pure cladus, you don't want to use that, because some of the descendants of a prokaryote would be outside of that classification, but it's still valuable. You can say the word prokaryote, and people will get a mental picture of what you're talking about, right? A cell lacking a nucleus, lacking any other membrane-bound structures, usually having a cell wall that's composed of either peptidoglycan or something strange in the archaeans, right? People have a mental image. When you say protist, nobody gets a good mental image of what you're talking about. It's just, it's a mess. It's the junk drawer, right? So if I told you, give me 5 characteristics of your junk drawer in your kitchen that's separated from every other drawer in the kitchen, man, it'd be hard. You'd be like, well, it's just a mess. So you could do that, right? If you, one characteristics of protist is, man, it's just a really messy grouping. And then you could maybe give another one and say it's any eukaryote that's not an animal, a fungus, or a plant. Right? But to come up with other unique features, that's an impossible task. And this is not the last time I will give you an impossible task for you to do, because it's fun. Sometimes I'll do it on exams. I will give you an impossible task, but I'll let you go with it, because you'll be equipped to get there. And then if you can actually figure out that what you're doing is an impossible task, and you point that out, that's wonderful. I love that.
And if you complete that impossible task,
Well, you can't complete an impossible task, then it's not an impossible task.
Okay,
Yeah.
so maybe a characteristic of them is that they don't fit nicely.
Yeah, or that we should just we should just ignore this way of classifying organisms. We should get away from having this group in textbooks, right? Which I think we absolutely should, but until it does, we got to deal with this, right? We've got to have a chapter, we got to have a week on protists, and we got to figure out how we're going to work on this. All right. We got a big task ahead of us right now. We've got 12 more slides to cover in 17 minutes. As an aside, we may be able to do it. I don't know. I don't really care all that much. My first time ever teaching at the college level, I would go through 75 slides every 55 minutes. And man, I just killed it. I'd get up there and I'd just be like, this, and this, and move to the next slide. And I'd just go. And then I realized it's not a great way to actually teach material. And so now I'm actually lucky if I could do 15 slides in 55 minutes. But yeah. So if you feel like there's a lot of slides we're going through, just know it could be a lot worse. And that's usually not a comfort saying, well, it could be worse, right? It's like, oh man, you know how horrible my day was? It could be worse. Of course it could be worse, but it could also be better, right? All right, so the next framing question: how are Giardia well designed for their lifestyle? And this is going into Excavata, the group of eukaryotes I told you we'd save for the end because of kind of some unique features. So Giardia, this is a genus. They are diplomonids that are in this supergroup Excavata. So some of their structures, some of their design, they have non-functional mitochondrial remnants. For a long time, we thought that they just lacked mitochondria entirely, and may actually be a snapshot of what the early eukaryotes would look like. Which I always thought, well, that's really absurd, because when you talk about the origins of eukaryotes, you always do it around the formation of the mitochondrion. And we'll get to there. We'll have that discussion. That's what you're doing for your homework. I always thought, wow, it was weird why you would think this group without a mitochondria would be our early group. I always thought it was just because my view of origins was different, so I was dumb and I couldn't understand it. But it really doesn't make any sense. And then on top of that, we actually found mitosomes, which are derived from mitochondria. So these organisms at one point in their ancestry did have mitochondria, and they lost them for a specific reason because of their lifestyle. Their lifestyle don't require it. So how are they well designed for their lifestyle? Well, one, they're very simple, right? They lack mitochondria, and we'll get to why they don't need it. Oh, right now. So they live in and parasitize vertebrate hosts. And so inside of this, they don't need mitochondria. Mitochondria are the powerhouse of the cell. They are what help the cell to generate the maximum amount of energy from every food molecule that they get. But these are tiny organisms living in the intestines of a really big organism. They are surrounded by so much food, they don't know what to do with it, right? And so having functional mitochondria is wasteful because it takes up space in their cells that they could divert to something else. And that is making as many offspring as possible. Because that's what parasites do. They are basically just reproductive bags. Okay? And so that's their goal. And so they don't waste the space with mitochondria because they have more food than they know what to do with. It doesn't matter that they're wasteful because there's plenty of food to just overcome that wastefulness. Right? It's like going to an all-you-can-eat buffet. And you eat something, you're like, well, I didn't like that. It looked good, but it's not good. And then you'll just like let it go. And you usually wouldn't do that with other food, but you're like, I got plenty of food here. Like, what do I care? But sometimes there'll be somebody in your group like me, and it's like, no, you can't. You can't throw that back. I'll eat it. So, but yeah, this is really interesting. And plus, the oxygen is really scarce inside of the intestinal tract of these hosts, and the mitochondria needs oxygen in order to get that maximum amount of energy from out of the food source. So not only is it wasteful to have the mitochondria because it takes up space that you can use for making more giardia, but it's also not helpful because what you would need to actually use them isn't there in very much abundance. They have several flagella that make moving through the intestines possible. Now, the nice thing about the intestines, if you're living in somebody's intestines, is you'll move. I mean, you'll move without having to actually think about it, right? Our intestines do a really good job of moving material through the intestines, right? That's why things don't get stuck. Usually, if things get stuck, it's bad, right? Things move through, but sometimes you have to move back upstream, right? You're like, I really wanted to be in the small intestine and not really in the large intestine because it's a little creepy there, right? And so you need to sometimes move back upstream, and they have flagella to help them do that. So how are they well designed for their lifestyle? Well, they basically have simplified everything that can simplify and invest in only what they need to live in the intestines and feed off of a host. And they are just incredible parasites. Here's an image of Giardia lamblia. Causes a disease known as Beaver Fever because you tend to contract it from drinking fresh water in contaminated pools. And a lot of times in most of North America, beavers like those kinds of freshwater pools. In fact, many of them make those pools by damming up, small little creeks. And yeah, believe it or not, it probably isn't hard for you to believe. There has, in the past, at this institution, been outbreaks of Giardia from drinking from the water fountains. Right? It hasn't happened in a long time.
Here?
Yeah, here. Where you're sitting. It hasn't happened in a long time, but in the history of this institution, people have gotten Giardia from drinking from the well here. Okay? Which is why when Dr. Jones was still alive, and if he saw anybody drinking from this water fountain, he would smack their hand away from the little part that gets it going and walk them to the water cooler in this little suite of offices. And he would say, "You're welcome, I just saved your life." It's actually, it's not pleasant to contract Giardia. You call it giardosis or beaver fever if you want to be fun. It's not devastating, but it's pretty close to devastating. Yeah.
What infected the well?
Giardia. Yeah, I don't know how it got there in the first place. I don't know. Yeah, I don't know. Sorry for answering your question that way. I could not help it. I couldn't help it. Did you have a question, Lacey? Okay. All right. Any questions about this? Yeah. Basically, the symptoms of eating, eating like, food that has been contaminated with bacteria. It's basically uncontrollable diarrhea, right? Lots of fluid moving through your intestines that you don't really want moving through there. Yeah.
How do you cure it?
You can take antiparasitics to kill the Giardia. Yeah, if you have a really, really heavy infection, it takes some time. It takes some time. So, yeah, but it's doable. Killing intestinal parasites usually isn't a big challenge because getting the drug there is not very hard, right? If you swallow it, it's going to get there. Treating parasites that are located in obnoxious places in your body, those are really challenging. So next framing question, why do body plans simplify with certain feeding strategies? Right, we just said how are Giardia well designed for their lifestyle, which is parasitic. Well, why does that actually happen? Why do body plans simplify with certain feeding strategies? So parasitic protists often do not have mitochondria, like Giardia. I mean, they have mitochondrial remnants, but it's not really up for very much debate now whether or not they came from an ancestor that had mitochondria. I think they obviously did, and then they lost that as they became parasitic and left a free-living lifestyle. You have another group of protists that are called saprobes, and these organisms, they feed on dead and decaying material. So bacteria, fungi, and protists play a huge role in that process of decomposition, unlocking that organic material. And so saprobes do that. And so you don't have to work very hard, right? What you're eating isn't running away from you, right? It's just sitting there. And you probably got there because you were carried by something like the wind or by some other organism carried you to that dead material. And so a lot of these organisms, parasites and saprobes, they oftentimes lose sense organs, the ability to detect and respond to light. What do we call that movement in response to light? Do you remember that? Taxis, right? So you lose the ability to do that if you can't detect that stimulus in the first place. And they oftentimes lose the ability to move incredibly well, except for as far as they need to. Also, many parasitic forms are small and reproduce at extremely high rates compared to free-living forms that are similar, that they may have actually come from. And this makes sense. The smaller you are, the faster it is to make copies of yourself. And then if you're going to lose the need to invest in things like mitochondria that take up space and invest in things like sense organs that take up space and energy, man, I mean, what else are you going to use that energy for? And you have an unlimited food supply. So you ought to use it for making copies of yourself, and that's what these parasites do. Parasites and saprobes oftentimes lack light sensing capabilities and chloroplasts. So you can actually trace some of these saprobes and some parasites back to photosynthetic ancestors. And legitimately, it works well. There's no question. There's no polytomy. It's nice and clean and neat. And it's very obvious that, yes, in fact, they did start as this free-living photosynthetic organism that then entered into a parasitic or saprobic lifestyle. Saprobic, eating dead, decaying material, helping that process, parasitic, feeding off of a host. And a lot of those things became not necessary, right? You don't need to be able to take sunlight and turn it into biomolecules if you're living in the intestinal tract of a host. Not a lot of sunlight penetrating to there. Yeah.
Because you're talking about how it's pretty easy to trace certain organisms back to another and how.
Yeah, when it's clean, sure.
Well, yeah, but how would you approach that from a creationist standpoint? Because that is a visible, tangible form of evolution. So
Sure.
How would you, from a biblical standpoint, how would you go about explaining
Right. And so there are a couple of ways to deal with that. One is you have to remember, Scripture is not a science textbook, so it's not going to give you all of the details about how everything works. Scripture also uses words like created after their kind, right? And so there's a question, well, what does that actually mean? Is kind synonymous with species? And I think very few people think that that's actually true. So for instance, kind probably represents something more of a specific pattern, a specific creation, and then the diversification happened after that. And so, for instance, I think it's almost universally accepted that all of your cat species originated as a single type of creation and then diversified. There's actually very, very little structural diversity among cat species. Most of it's behavioral differences and then maybe some color pattern differences, which is easy to accomplish. And then so what it looks like is you do have limited amount of diversification possible. And we see speciation happen. So like if you took fruit flies and you took them to a new island that they had never been before, and you just let them go, right, and let them work whatever, especially if people live on that island, and so they're harvesting fruit in different ways than animals are harvesting fruit, within a couple of generations, which the generation of a fruit fly is like 5 days, within a few generations, you're going to have separate species. I mean, it started with one, and then you're going to have things where you have legitimate reproductive isolation, you've got some structural diversity, and you can actually classify them as separate species. So this happens. We see speciation happen, meaning that it has to have always happened, and then it's more of a question, well, what are the limits of that, right? Is speciation the same thing as trying to root all of your eukaryotes back to a single ancestor? And it's not even close to the same thing. Right? Speciation is just basically a byproduct of getting some change in allele frequencies. And we're going to talk a lot about this. We'll talk about the mechanics, the mechanisms of evolutionary change, and we'll talk about, well, what does that mean and where are the limits? And I'll give you a nice analogy. I didn't come up with it, therefore it makes sense. The analogies I come up with, if they do make sense, they revolve around food. But I don't have a food analogy for it yet. But yeah, we'll deal with that. Yeah.
So, like, in other words, you would say, like, it's more like God created, like, a cat, and then, like, God created, like, a dog. The cat gave rise to, like, bobcats, lions, house cats, and the dog gave rise to, like, wolves, dogs.
Yes.
Is that, like, how you would say
Yep.
it?
Mm-hmm.
Okay.
Yeah, and I think you can see the discontinuity. So if you take all of your species of dogs and you compare them with other life forms, you'll see where that discontinuity is. Where you have a nice smooth transition from one form to another, where it's clean. When you try to build them all and root back to an ancestor, you don't end up with polytomies. You don't end up with unresolved relationships. You don't end up with, really weak, where it's like, man, I built this tree 5,000 times, and only in 100 of those did this branch separate by itself, right? That's horrible. That means in almost all of the trees you built, this branch was going somewhere else. And you're like, well, how much confidence do I have then in putting this kind of a tree together? See, when you do it to where what you're modeling is actually the trajectory, is actually reasonable and manageable evolutionary change, it's usually clean. Yeah.
So what would we call, like, if we can't call it species, what do we call, like, all the different kinds of cats or all the different kinds of dogs? What's the term we use?
You would use a kind, like the cat kind or the cat type.
Okay.
Yeah, I mean, Scripture tends to use that term. Brahman is the term. This kind, this original created form. You could also call it form or unique creation. Yeah, and I think we can see evidence of that, and we can see where that discontinuity exists, and where we can see that there are issues in trying to root them all back. Yeah.
There is evidence of speciation that we can see in our world.
Yep.
That evolution can take and manipulate.
Well, sure, you can take and extrapolate it and say, hey, if it can work on a small scale, why can't it work on a large scale?
Yeah, but
Sure.
but there hasn't. So there's evidence of speciation,
Uh-huh.
but there's not. Is there actually evidence of a form of change of kinds? And if
No. Yeah.
well, then why is it still taught as a possibility even if it hasn't?
Well, the question is, I mean, when you're dealing with millions or hundreds of millions or billions of years, who knows what's possible, right? We have 200 years worth of good data, right? We have 200 years worth of reliable, well-structured, well-kept data that was recorded without, enormous bias. Well, how do you then compare that to billions of years worth of time? I mean, you just can't.
So do you think that they can use the time frame of billions of years as a form of a cop-out?
Not as a cop out, but as an explanation. Yeah, I mean it's a legitimate tool. I mean billions of years. That's a long time. That's a long time. All right, it's past time. It's not my fault. Have a wonderful day. Enjoy the rest of your Wednesday.
So, polytomies. I have to ask.